Okay, so now that we know how T cells are activated and how B cells are activated, we're now just going to discuss interactions of T cells and B cells, or T cells and other immune cells. And so, just as a general overview, we have T cell help for B cells, and we have T cell help for other T cells. And so when we talk about T cell help for B cells are a presenting cell, and they are able to interact directly with CD4 T cells. So a B cell will be able to express MHC class II. And so a CD8 T cell, on the other hand, cannot express MHC class II. And so it's going to be different how a CD4 T cell interacts with each of these. A CD4 T cell, of course, can interact directly with the MHC class II, and it does so with its T cell receptor. And it's going to interact with some antigen that the B cell is presenting, the peptide antigen. I'll just draw it like a box. And so with this type of interaction, your CD4 T cell can give the B cell help directly. So it's a cell-to-cell interaction. At the same time, your CD4 T cell can also give a cell-to-cell interaction with a macrophage. The macrophage also can express MHC class II, and the macrophage will put in a peptide that the T cell can recognize, and it will be the same peptide because the specificity has to be the same. So CD4 T cell can also give help to the macrophage. Now a CD8 T cell over here again cannot express MHC class II, so the CD4 T cell cannot interact directly with the macrophage. But what a CD4 T cell can do for your CD8 T cell is, we'll draw it on this side. We'll have the MHC2. We'll have our CD4 T cell. And it's interacting with this MHC2. With that green peptide again. And this T cell, CD8 T cell, is being activated, we'll see over here, by a dendritic cell. And this is an MHC class I with the T cell receptor for your CD8 T cell. It's a bad T cell receptor. This is the T cell receptor. And this is the same pathogen, so it's the green pathogen, but the peptide might look a little different. And this is on MHC1. And so the DC, as we know, can activate your CD8 T cell. And your CD4 T cell can help by giving IL-2. And then IL-2 will bind to the IL-2 receptor. The IL-2 receptor is made up of a gamma chain, that beta chain, and the alpha chain, which is CD25. So this is the IL-2 receptor. And the IL-2, we'll make them pink dots, binding to the IL-2 receptor is the signal. For the T cell to divide. So the interaction between the CD4 T cell and the CD8 T cell is indirect. It's through cytokines. And I'm just showing IL-2 here. It could be other cytokines. The interaction and help between a B cell and a CD4 T cell is direct through MHC class II. And the interaction between CD4 T cell and a cell like the macrophage to get help is also through intracellular reactions. Now, the B cell is an antigen-presenting cell, and there's a reason for this. The B cell needs to receive help through the CD40/40 ligand. So, on a cell-to-cell basis, CD4 help is via CD40 ligand, which is also known as CD154. Now, this help for a B cell allows the B cell to do isotype switching and somatic hypermutation. The help on the macrophage will allow it to be a better killer cell. Our macrophages are good at killing bacteria as long as those bacteria and even viruses don't have virulence factors. As soon as you get a pathogen that has a virulence factor that can overcome a macrophage's killing ability, it really relies on a CD4 T cell to recognize that it's still presenting pathogen antigen, and the CD4 T cell will tell that macrophage, you're still infected, you better start killing whatever's inside of you. And it does it through that CD40/40 interaction. And so you can see that both the B cell and the macrophage are expressing CD40, and it's the CD4 T cell that expresses the ligand for CD40 in order to provide help. Now, the help for the B cell is going to be in the form of cytokines. So we're also going to get cytokines. And this is basically going to depend on what type of CD4 T cell this is. Is it a Th1 versus a Th2, etc.? And so not only does the CD4 T cell help the B cell in order to do isotype switching, it's going to tell it what isotype it needs to be. If it needs to fight a intestinal worm, it will tell the B cell, you need you to make IgE. If it's going to be able to defend against a virus, it's going to tell it to make IgG, for example. And so CD4 T cells, again, can interact with only the 3 cell types that make the class 2, B cell, dendritic cell, and macrophage. Its interaction with the dendritic cell is usually for activation, whereas its interaction with both the macrophage and the B cell is not only for activation, but it's also for providing help to those cells. So let's talk a little bit more about B cell activation and help. So we're going to do this from the perspective of a lymph node. And so I'm just going to draw very vaguely the lymph node areas. So we have here, we'll have a B cell zone, the cortex, and then we'll have an inner zone, which is the paracortex. And this is the T cell zone. And then we'll have the medulla. Now, there are going to be inferent lymph, efferent lymph. There are going to be blood vessels. All of that's going to be in the lymph node as well. But I'm not going to draw all of those. Now, with respect to the T cell zones, some things that are important are the T cell zones always have the high endothelial venules. From histology, hopefully you know what this is, high endothelial venules. And this is where you have your lymph. And this is where you will have your B cells from your lymph. And they will migrate to the B cell zone. You will have your T cells from the lymph, and they will stay in the T cell zone. And you'll have your dendritic cells from the lymph, and they will enter the T cell zone. So B cells will kind of go into their own little area, and this is through different chemokines that we'll talk about in future lecture. And you have a B cell zone. Your T cells will interact with your dendritic cells. So you'll have dendritic cells that express both class 1 and class 2. They will have a peptide antigen on these, and they're different sizes. Class 2 is longer than class 1, and they will interact with different T cells. So you can have a CD4 interacting with the class 2, and then you will have a CD8 interacting with the class 1 with their T cell receptors. Now this is everything that we know. Now what happens with this antigen is in the B cell area, you're going to have a special class of macrophages that line the sinuses. So here's a macrophage. And these macrophages are subcapillary sinus macrophages. I'll write that. And these macrophages will have different receptors on their surface, but you can kind of think of them as a pincushion. So these are not going to take up antigens. Instead, they're going to have like these sticky little fingers. And on the sticky little fingers, it's going to have different shapes of antigens for your B cells to see if they're specific for. And so the B cells will sample these sticky fingers on these subcapillary sinus macrophages, and some of them might be specific for one. So let's just say that this B cell here has a B cell receptor that will bind to that little circle. And so this B cell will now be selected. So this B cell, I'm going to start drawing as a red one because that's the color of the antigen. So this B cell will proliferate. And the other thing this B cell will do is this B cell will migrate to the junction between the T and B cell zones. And so you'll have this B cell here, and on its surface it will have MHC class 2, and it will have its little red peptide in MHC class 2. And the CD4 T cell will migrate to the B cell T cell junction, and this CD4 T cell will interact with this peptide and will give help to the B cell. Now a few things are going to happen with the B cell, and it's a process that's very involved, and this is why it takes a long time. So to go from an IgM, I should put it here, these guys will be making IgM. This guy will be making IgM until after help. I don't want to confuse people. But to go from IgM to IgG or A or E takes about 2 weeks. So it's a long time. So basically what's happening is this B cell is now going to migrate to the medulla. And this is going to be the primary focus. And the B cell in the medulla is going to divide and replicate. And some are going to become plasma cells. And these guys are going to make IgM. And so this is going to be short-lived. And some of these are going to go back to the B cell zone and become a secondary focus, and they're going to form a germinal center. Now in the germinal center, you're going to have a lot of what we call blasting cells. So they're very dense. So the B cells are dividing like crazy. So you're going to have blasting. And then you're also going to have regions in your germinal center where you have your follicular dendritic cells. And follicular dendritic cells are going to be similar to those subcapsular sinus macrophages, and they have sticky fingers. And you're also going to have in here, you're going to have your follicular helper CD4 T cells. So you'd have some CD4 T cells in here. T follicular helper cell. And then these are all going to interact with your B cells. And the B cells are dividing in that dense area. And in this more spread out area or light zone, you're going to have the interaction of the B cells with CD4 T cells and your follicular dendritic cells. And from this light zone, you're going to get your B cells that switch and now make IgG, for example. And these can go to memory, or they can go to long-lived plasma cell. Okay, so all of this has to happen for B cells in order to become a memory cell and to also undergo isotype switching. So really what's happening in the germinal center, so let's do a really quick germinal center reaction. So for the germinal center reaction, you're going to have a dark zone, and you're going to have a light zone. And so in the dark zone, you're going to have centroblast. And light zone, centrocytes. And so the dark zone is basically massive proliferation. And it looks dark because the cells are basically on top of each other. And the germinal center kind of looks like a circle. So it kind of, if you look at a lymph node, it'll kind of have a circle, and this will be the whole germinal center. And so your dark zone, where your cells are proliferating, are going to be all around this area, because there's a lot of proliferation going on. And then in the middle, or in some sort of area within, you're going to have your follicular dendritic cells. And why this is called the light zone is because of the other cells present, especially your follicular dendritic cells, you're basically going to spread out your B cells. And you have some CD4s in here too, so these are your 4s. And they're going to provide help, continuous help. And your B cells just get stretched out. They're looking for antigen on the surface of your dendritic or your follicular dendritic cells. So there can't be as many. So this will be the light zone. It's going to be over here. And so from the light zone is really where you're going to get your good effectors. they're going to form memory, and they are also going to form those long-lived plasma cells. Okay, so as I said before, CD4 T cells are going to help your CD8 T cells indirectly. Now, some things you always have to keep in mind when you think about CD4, CD8. CD8 T cells are cytotoxic. They can't be anything else. And so they are really good with intracellular pathogens. And so the T helper response that's really good for intracellular pathogens is the T helper 1 response. A T helper 2 response really can't help a CD8 T cell because the T helper 2 response gives the wrong cytokines. So T helper 1 response, you will have your, we'll set up the TH1 environment. So TH1 environment from the innate, remember the innate is the alarm system, is IL-12 plus interferon gamma. So let's pretend we have a virus infection. So here's our epithelial cell, and we're going to have our coronavirus infect our epithelial cells. Here's our coronavirus. Our macrophage, here's our macrophage, will release IL-12 in response to the coronavirus. It'll also pick it up. And IL-12 will trigger our natural killer cells to make interferon gamma. So IL-12 plus interferon gamma is going to lead to a TH1 helper cell. So basically, you have your dendritic cell. Let's draw our little dendritic cell. And it's going to interact with our CD4 T cell, or CD4. There's a T cell receptor. And your dendritic cell is also going to interact with your CD8 T cell. And then the dendritic cell, of course, is going to display the coronavirus peptides on the surface, and they're going to get activated. The dendritic cell plus the IL-12 plus interferon gamma will tell the CD4 T cell you need to become a TH1 cell. And then this TH1 cell will release IL-2, it will release interferon gamma, and this will help the CD8 T cell be able to become a better killer cell. But it's only the cytokines that are helping the CD8 T cell. Okay, so B cell help is pretty straightforward. We've already talked about it. We talked about macrophage help just very quickly. You have your B cell that's activated. Now, one thing to keep in mind is the B cell on its B cell has the B cell receptor on its surface. Let's see, it's square. I'm going to get rid of this part because this is taking too much space. It's going to have a a antigen on its surface. And basically, the B cell is going to internalize this whole complex, chop it into pieces. So you'll have the B cell receptor, you'll have the antigens in purple. This is going to fuse with a vesicle that contains MHC class II. And so you'll get fusion of the two vesicles. So you'll have these peptides. Some will be self, some will be virus, and they get loaded on the MHC class II. MHC class II goes to the surface with this virus peptide. And yes, of course, some of it will go to the surface with the self peptide, but hopefully you eliminated all those self T cells, and the T cell specific for this antigen will get activated. So then you have your T cell come along, and it's going to see this antigen on the surface, and it's going to become activated. So this is signal 1. It's then going to have CD28, and your B cell will have CD80, beta 7, whatever you want to call it. And then you're going to get the helper signal, which is your CD40 ligand, which is CD154. And this is going to bind to CD40. And so this is the help signal. And this is signal 2. And so all that happens, nothing has changed. And what the help signal does is the B cell is now able to undergo isotype switching plus somatic hypermutation. And all of this occurs in the germinal center reaction. Now, somatic hypermutation is basically where you have your B cell receptor. So here's your B cell receptor. And this area is the variable region. And if you blow this area up, here's your variable region. You will have in it these areas called constant, and then you'll have in it these hypervariable regions. And it's these hypovariable regions that change. And so they just get better at binding to antigen. So I've shown this before, but just really quickly, this is pre-somatic hypermutation. You have an antigen binding site that looks like this, and your antigen might look like, say it looks like this. Here's your antigen. And then after your somatic hypermutation, your antigen binding site looks more like it will fit your antigen. And so the antigen fits better inside. So it becomes a better fit. Okay, so that's what B cell help is doing, and it's coming from the CD4 T cell. And the other thing to note is that B cells, even though they have the ability to activate CD4 T cells, B cells present specific antigen 100 times better than other antigens, than nonspecific. And that makes sense because the B cell receptor is on the surface, it's grabbing the antigen and it's internalizing it. It doesn't really grab nonspecific antigen, so it's not really going to present nonspecific antigen. And so it's really the CD4 T cell that's specific for the same antigen that the B cell is specific for is going to help that B cell. That's how you kind of control a flu B cell will be helped by a flu-specific CD4 cell, and a cold B cell will be helped by a cold-specific CD4 B cell. A tuberculosis B cell by a tuberculosis-specific B cell. And so that's how they make it that specific. Now, macrophage help, again, just very quickly goes through CD4 MHC class 2. And so in this case, you have a macrophage. It has an endosome, and it has bacteria inside. So here's our bacteria. And say the bacteria has virulence factors, so it prevents fusion of your endosome lysosome. So the macrophage, it will have a lysosome compartment. Make it pink. Here's its lysosome, and it has all those nice digestive enzymes. But it's not fusing with the phagosome or endosome that contains the bacteria. And so in this case, you have on the surface again, MHC class 2. You have interacting with this again, your CD4 helper. Let me bring these guys all down. Okay, we have our CD4 helper. And so the T cell receptor, and then you're going to have the same sort of signals. You're going to have the CD80, 86, beta 7, and this is going to interact with your CD28. Hasn't changed, signal 1, signal 2. And then you're going to have your CD154, and that's going to interact with the CD40, so CD40 ligand, and this is going to give the help to the macrophage, and it basically tells the macrophage, hey, you're infected. So here's the peptide from that red bacteria, and so basically after it gets this help, the macrophage, here's our macrophage, and it'll cause fusion. So now you have the endosome fusing with the lysosome, and now your bacteria that were inside are going to get digested into peptides because of all the digestive enzymes that were in the lysosome. And your T cell basically helps your macrophage kill the pathogen. All right, so some intracellular pathogens. We have extracellular pathogens, and we have our larger hemolymph type pathogens. And so they're all going to require different types of helper responses. So the most common are going to be viruses. So viruses are the most common pathogen. So our most common immune response because of that is going to be our Th1s. Now, there also are some intracellular non-viral pathogens like Mycobacterium tuberculosis. Many people are familiar with that particular pathogen. It leads to a chronic infection. There are also some protozoas and fungi that are intracellular, and Th1 responses are best against them as well. The CD4 cytokine that was produced is interferon gamma. The innate input is going to be macrophages, and macrophages are going to create IL-12, and NK cells are going to give you that interferon gamma initially to create that environment. They're going to activate your cytotoxic T cells as well as cause isotype switching to IgG. And you have a master transcription factor, which is TBX21. And so this is basically turns on Th1 genes. Now, if you have your larger ones, your hemolyths, your indicators of the response are not known. It's generally thought that you have to create a Th2 environment, which means you need IL-4. There are many cells that can produce IL-4. Some endothelial cells can, some epithelial cells can, mast cells can, basophils can. So it's probably a combination of those cells that create that initial environment. The Th2 cell is going to be activated. It's going to release a different series of cytokines, IL-4 and IL-5. These your CD8 T cells don't respond to, so it's not going to activate your CD8 T cells. Instead, you're going to get different cells, mostly your innate response is going to be activated. Now, also, your B cells are going to produce IgE, and your master regulator is GATA3, and it turns on Th2. And then you have your extracellular pathogens, primarily bacteria, some fungal elements such as Candida. You're going to have your macrophages make IL-23. This is going to induce a Th17 response. Th17 cells make IL-17. This is really good at neutrophils and antimicrobial peptides secreted by epithelial cells, for example. Neutrophils are really good at coming in and eating extracellular stuff. They're not good at eating intracellular stuff. They can't see it. So they come to the site of infection and they will eat whatever is there, bacteria, fungus, whatever. The adaptive immune system, you really don't have much going on. I don't like this too much because you can get IgG, and IgG is going to be helpful in helping your neutrophils and phagocytic cells eat the extracellular bacteria better. ROR gamma is the transcription factor that turns on Th17. And so these three transcription factors are really epigenetics in real time. If you have a virus infection and you turn on a Th1 response, you in essence are blocking Th2 and Th17 responses within that same helper cell. So in essence, your T cells are able to manipulate the transcription of their genes based on real-time inputs from the environment that they're in. And so that's a really good example of how epigenetics works. Okay, so let's just go over a little bit about these. So Th17 is good for extracellular. And so you're going to recruit newts plus your innate defenses. And so in general, really, your antibiotics are very helpful, or anti-fungal agents are very helpful for this. For Th1, you're going to have your intracellular pathogens, and you're going to have a Th1 response, and critical are CD8 cytotoxic. Th2, you're going to have your mast cell plus eosinophil. And again, these are going to be usually too large for your T cells to even handle. And so you need mast cells and eosinophils. Eosinophils have major basic protein. And they release major basic protein, and it punches holes in larger, like worms and parasitic infections. So this can digest, for example, a worm's outer layers. And mast cells, of course, has histamine and a whole other bunch of factors, and it helps with getting worms expelled from your digestive tracts. It causes the contractions of your stomach muscles, etc., that help to expel those worms from that. And so really the type of immune response you need is dependent on the type of pathogen, and it's very important to get those correct, because if you don't, then you can potentially have the wrong immune response for the wrong pathogen. A classic example is Mycobacterium leprae. So if you have a Th1 response, you control the bacteria. If you have a Th2 response, you succumb and you die from the bacteria infection. So again, it's really important to have the correct response for the type of infection. Now, I just want to talk about a few other things because we now understand T cell help. And so there are conjugate vaccines, and a conjugate vaccine is basically tricking the immune response. So I already told you that a B cell will be able to pick up antigen that is specific for and present it to a T cell. So there are a few things you need to know. One, a B cell can respond to antigen directly. Two, a T cell can only respond to a peptide. And so what that means is if you have a B cell, that on its surface it has its B cell receptor, and it's responding to this antigen. Oops. Oops, it keeps doing that, sorry. It's responding to this antigen. And if this antigen is a polysaccharide, too many C's, then your T cells cannot get activated with a polysaccharide because a polysaccharide does not become a peptide. It's basically a sugar. And so in order to get your B cell help, so the scenario would be, here's our B cell. Your B cell will release IgM, no help, no germinal center reaction, no isotype switching, no somatic hypermutation, and no memory. So in essence, for some bacteria, for example, you would have a primary response every single time you get encountered with that particular bacterium. Because a B cell is only recognizing as polysaccharide and it can't get help from a T cell. And so a conjugate vaccine, let's put this in its own little box, takes a protein plus the polysaccharide and tries to induce immune response. So basically, you are going to take this polysaccharide and you're going to tag it with a protein, tetanus toxoid, let's say. And the B cell that is specific for that polysaccharide, so here's our B cell, it's binding the polysaccharide. It's going to internalize not only the polysaccharide, but it's also going to internalize the protein. Oops. And so on its cell surface, the B cell will express MHC class 2 for the protein, and then a T cell, a CD4 T cell, that is specific for this protein, the tetanus toxin, for example, will bind and give help. Now this B cell was not specific for tetanus, and so this B cell instead is going to release IgG of the polysaccharide. So you're basically using a tetanus-specific CD4 T cell to provide help to a polysaccharide-specific B cell. And so it's a way to kind of trick the immune response into responding and having memory against polysaccharides on some common bacteria. And we also, you guys might have heard of the Hib vaccine. And this is Haemophilus influenza B vaccine. This is a conjugate vaccine. So the vaccine does work. And Haemophilus influenza type B was a common cause of meningitis in teens especially. And now that we have this vaccine, pretty much we don't see any meningitis caused by Haemophilus influenza type B in the United States anymore. And it used to be one of the leading causes. Now the last thing I want to talk about is recurrent Candida infections. This is a fungus, common fungus. It is a normal flora fungus. And so if you have a patient who gets Candida infections over and over again, especially in the mouth, so in the mouth it's called thrush. And this is an indicator of a T cell or an immune deficiency. Now, Candida is everywhere. So it's in the environment. You can't get away from it. And our immune system is very good at defending against it. So even though we don't know everything about our immune response to fungal infections, CD4 T cells, Th1, that are producing interferon gamma, or Th17 that are going to recruit neutrophils are typically the best protection during fungal infections. And so if you continuously have repeated fungal infections, it's an indicator that there is something wrong with the T cell compartment. Because even if you need antibodies for this, you need your CD4 help to get those antibodies to help you against this fungal element. The fungal elements are typically extracellular, so it's not your CD8 compartment that's usually involved. So it's going to be usually your CD4. Okay, so that's all I wanted to talk about today.